so let's talk about our last section, which is lacto operant. I always want to spending lots of time talking about the lacto operant because you learn in your other classes, but it's like a base for some of you. So the lactose, or the glucose, there's one is attached, one is not attached, one is transcripted, one not transcripted. It's like a maze. The reason is there is a lot of the knowledge behind it, and it's very comprehensive. So that's why we want to talk at the end of this section, and also it's some knowledge related to we learned in our exam 1 even. So we want to spend some time to talk about that. Okay, so here is the thing. In this section, we talk about the metabolic process. Okay, we have a glucose goes to the fructose 1,6,5-phosphate, then go transition step, become acetyl-CoA, and then we had a branch here and generate pyruvate through glycolysis, and this pyruvate we could go through fermentation, and they also could go is a transition step become acetyl-CoA, and then we'll go through a TCA cycle, citric acid cycle. And then we also talk about the central dogma, and we talk about DNA replication by itself, and we also talk about RNA transcription, and the last one we talk about is RNA become amino acid and then become protein, which is translation. So this is too big picture of the outline of this exam. So we can start to talk about lacto operant from here. Okay, so for the bacteria, they need to survive. They have to go through these central metabolic pathway. And this central metabolic pathway, they need basically the enzyme to make sure these reactions happen efficiently. And these enzymes, the major components is actually a protein. So that means they need a replication, transcription, translation to do constantly and immediately and efficiently. So for a normal bacteria to survive, they have some of the gene to control these in a very efficient way. And those type of the gene, we call it a housekeeping gene, or we call it a constitute genes. Another good example will be, we talk about the cell wall structure. Bacteria have to biosynthesize peptidoglycan. If you synthesize peptidoglycan, those gene is also a housekeeping gene, because bacteria to survive have to rely on these important gene expression. However, they live in the environment. It is different from what they live, let's say, in a bacterial medium, because the environmental situation will be changed. So what happens is, let's say there is a light coming. Okay, this is like a stimulant. And then bacteria have a cell wall, cell membrane system, and the cell membrane system basically is a sensor. And this sensor have to be amplified, this signal, and then will be regulated. Some of the gene will be open and some of the gene will be closed, to find an economical way to survive. This is what we talk about is gene regulation. And there is basically two levels of the regulation. If the regulation happened related to the mRNA, we call it transcriptional regulation. If the regulation relates to the protein and the amino acid, we call it a translational regulation. So this is what we talk about that. Now, why we want to talk about the lacto-operon? Because the lacto-operon is the most well-known studied gene regulation system, and this is based on the E. coli. And the bacteria everybody knows. So that's why we want to talk about it. That's a big story behind the background. What is lacto-operon coming from, okay? So we move these off, and then we use this whole blackboard. We want to explain these things to you, the detail about the lacto-operon. Now, before that, we need to give you some extra background information. So this is called the gene regulation and related to the structure genes. So we would like to draw here. So the gene regulation. So here is what we call the transcriptional regulation. So transcriptional regulation, we need to talk about the structure gene. The structure gene could be complicated, could be relatively simple. We make sure it's a relatively easy, okay? We will be split these onto a couple boxes. I will be first to start with these 3 boxes. These 3 boxes, we gave them a name, is A, P, and O. What is A? This is activator protein binding site. What is O? This is operator. Then what is P? It is promoter. So let's review about it, what we learned in last class. What's a promoter for? It's a very conservative DNA sequence. Will be ideally coupled, attached with a polymerase. We call it is RNA polymerase. If they attached together, then what happens? We'll do RNA transcription from 5' to 3'. And at the end of the day, the right side, this is a functional gene, will be transcribed, become mRNA. So let's review what is a promoter. Do you remember -35? TA, GA, CA, that's a conservative sequence. Do you remember -10? TA, TA, AT. Is that right? That's called the promoter box. And the RNA polymerase, you need to recognize this very conservative sequence. Then they can be attached with each other. Now, RNA polymerase is a hollow enzyme. They have 2 alpha units, beta prime, and omega. That is called coenzyme. And how they recognize promoter? They need a sigma factor. And most of the function, we need a sigma 70, and that's combined together is a hollow enzyme. So this is something we already talked about. Okay, and this is the structure of a functional, of a structure gene. Now, we want to mention something, introduce something to you. What happened? So-called positive control, negative control. So let's draw here again. Okay, we have 3 locations. A, P, O, and this is a RNA polymerase. So say it very simple. Maybe it's confusing you in different description. Everything happened we split this onto this section. Okay? Everything happened here. We call it downstream. And also said very simple. Something happened for this O position. This is negative control. In other words, if anything happened for the upstream, and something happened for this A position, now of course A position have a different name. This is called a positive control. Now both positive control and negative control, they could be separated into two different categories, which is inducible and repressible. So I always give a joke, then you can understand it very well. The RNA polymerase combined with the promoter, it's like two people get together, start from dating to get married, and they have a different pressure. That could be happened from their parents' part, which is we call it a positive control. They could have some pressure from their in-law parts. That's a negative control. That's exactly what happened in your lifetime and what happened for the bacteria. Now, from each of their side, they also need to have some inducible or repressible. That could be the money issue. That could be some of the education for the kids, for some of the other issues. These type of things could be inducible or repressible. So what happened is all these things will put a lot of pressure for the RNA polymerase and the promoter to get married, to get together. But the bacteria have to solve the problem. That's what it is. Otherwise, they will be dying. So you have to combat all these pressures, and at the end of the day, they can survive. So this is what it is about. If we give you a joke to express it. Okay. Now we're going to draw the picture to explain to you what is called negative control inducible, repressible, what is called positive control inducible and repressible. Then we go talk about lateral output. So we need to draw several, usually we draw four, okay. We'll just draw four structure right here. Every single thing, we do the same thing. We draw it. A, P, O. Let's talk about this guy first. So, first of all, let's talk about the negative control inducible. So, what is that? This means originally, this O position, the operator, had something attached to it. This is, we call it a repressor. Some repressor is in. Because they are attached to the operator position, so RNA polymerase, this is RNA polymerase, cannot attach to the P position. Therefore, the 5' to 3' transcription cannot happen. Okay, I want to solve the problem. So what are we going to do? There is some of a chemical needs to be there to attach to the repressor. And this is called an effector. And this effector is an inducer. And once this inducer attached to it, will change the morphology of the repressor and will let them release. So what happened is, if you use an eraser, they will be released and not attached to the O position. Therefore, RNA polymerase and the promoter will be get together and the transcription will be happening. At this moment, we call it negative control inducible gene. What are the good example for this? Lacto-operon is a good example. We will talk about the detail. Okay. Second example we will talk about is negative control repressible gene. Repressible. So still position A, P, O. So what this means? Originally, this repressor is not attached to the O position. So, RNA polymerase attached to the polymerase attached to the promoter and the transcription to 5' to 3' is happened naturally. However, I have some effector goes there. That's an effector. Attached to the repressor. And it become a friend of that, so we call it a co-repressor. And once they work together, this guy will attach to the O position. Therefore, the RNA polymerase and the P will not be attached, and the transcription will be terminated. That's called a negative control repressible gene. What is the example for that? Trapto-operon. So this is a very smart way to work in for the bacteria. I don't have enough tryptophan, so go ahead to generate the tryptophan. But once the tryptophan has already been enough, stop it. I don't need that you generate more tryptophan. So at this moment, this more tryptophan become a co-repressor. They attach to the O, then the transcription stops. So no more tryptophan generated. So what's the difference between these two? This is actually catabolic process, is that right? This is anabolic process. And I don't want to spend too much talk about tryptophan operon because it's out of our skill. You learned in your biochemistry class, but just briefly mention. Okay? Now we're going to talk about positive. What is positive? APO. And we said negative control is anything happens for the O position. Positive control obviously is everything happens for the A. So what happened? Originally, I will say this is an effect. There is an effector there. They are not attached to the A. And I'm coming, another effector is called the inducer. And this inducer attached to the effector, let the whole thing attach to the A position, and brings RNA polymerase to attach to the promoter. Therefore, the transcription starts to happen from 5' to 3'. At this moment, we call it is positive control inducible gene. What is the example for that? Maltodextrin. We don't want to talk about that. We skip it. Okay. Okay. We left one. Didn't talk about that. APO. Still positive control. Something happened for the A. So what happened? Originally, this guy is attaching to it and brings RNA polymerase to the promoter, and the transcription happens. However, because of this repressor morphology of the effector, they stopped to attach to the A position. So RNA polymerase did not attach to the promoter, and this transcription stopped. I don't have a very good example for that, but if you learned arabinose is actually a circle. Okay, something happened in here, and in here. You learned in the other classes, which is a combination of both positive and negative control. That could be a good example to talk about. So these are four basic models to talk about the post-transcriptional gene regulation. We don't want to talk all of these very detailed. We want to use a lacto-operon to talk about the top one. Okay, so we remove these. We remove all these. Okay, we remove the whole section. We're going to folks talk about the lacto-operon. I also want to make sure this is gone, because I'm going to draw the structure right here. So I move this part out. So we're going to folks talk about lacto-operon. Lacto-operon is discovered in E. coli by two French scientists, Monod and Jacob. This is about 2013. This guy I forgot. Very long life. Jacob is Monod graduate students later on. When he got a PhD degree, they start to work together. It's a Nobel Prize in like 1970s, later 1970s. It's a lacto-operon. Okay. What's a lacto-operon looks like? Let's talk about something easy. Okay? Start with something easy. There is a three position, which is called CPO. What is C? Catabolite activator binding site. Some textbooks said catabolite activated site. Same thing. P, you know, promoter. O, you know, operator. There is a three functional gene downstream. What are they? They called LAC Z, LAC Y, and the LAC A. What is LAC Z for? Beta glycosidase. What is LAC Y for? This is beta galactosamines. What is the last one? This is called transacetylase. But I will say, we just say it is unknown, the function. Lots of the people say this is unknown. Okay? So, sorry, this is the structure. That's what the structure looks like. You look at the slides, a little bit complicated. There's a lack I, whatever. We just focus on those three. Okay. Once Monod and Jacob, at the early stage, what they find is everything happened in the operator. So what happened is, originally, there is a tetramer which is connected to the O place. Okay, what it looks like, this is a tetramer. They actually connected to the O is two different structures. It's connected to O1, O3. They And they also, sorry, no. They also could have connected O1 and O2 like that. And this tetramer, why they can do that? Because this tetramer have a very special structure, is helixy turn helixy. It is a secondary structure of a protein. Looks like that. And obviously you see the operator position. You have O1, O2, O3. O1 is very important. You have to attach it to stop it to do the work, to stop the transcription. And I want to also tell you is when we draw like this, it's a standard figure to let you understand. But actually in the real life, because DNA is actually twisted. So if you really want to draw the picture, it is not like that. It is kind of like the pins in the middle. This is O1, this is O2. Then you have O3 right here, other things are right here. Okay? That's why when we draw the picture for the tetramer attached to it is like a twisted. But here we don't talk about that too complicated. So originally this tetramer is attached to the O position. Therefore, this RNA polymerase is not attached to the promoter, so the 5' to 3' transcription is stopped. Therefore, beta-glycosidase permease, and the LAC-A, they are not transcribed. So they just stopped it. And this is a situation, what happened is no lactose. What happens if there is a lactose? If there is a lactose there, what is going to happen? They will have some other chemical comes up, which is called allolactose. Those allolactose is like an inducer. The inducer will be attached to the tetramer and change the morphology, so this is not going to attach to it. Which means here you're not going to attach, So what happened? RNA polymerase will be attached to the P position, and then the transcription starts to happen. So